give some idea about the concepts that we're using. Okay. Now, let's see. Let's do a couple of things here. I've got a laser pointer. Right. So, what causes IBD or the IBDs? Historically, we've always taught and thought like this, that there's a number of things here. One is it is a genetic disease, and I'm going to spend a lot of time talking about that tonight. The microbiome is important, and there are environmental triggers. And I'm not going to spend a lot of time on the microbiome or environmental triggers. I'm going to touch on them briefly. And I'm a very, very big believer that researchers in these domains should not be working in silos. We should be working together. So it's never a competition between myself studying genetics and my colleague down the corridor who's studying the microbiome. We bring these together. It's the interaction of these things, as you can see from this Venn diagram, that ultimately leads to IBD. There are a few exceptions to that, and I'm going to show you some of those. So we've known that there's a familial or genetic basis for these diseases for a long time. This is Joseph Kirschner, who himself had great genes. He must have done. He was still practicing medicine over the age of 100, still seeing people with IBD in his clinic over the age of 100, but unfortunately passed, I think about 10 years ago now. But what you see on this figure here is a family, what we call a pedigree. And so these are, we could say, the children. These are the parents. This is the grandparents, and this is the great-grandparents. And as you can see, as you go through this generation, there's a strong family history of, here in orange, Crohn's disease, and this one individual has ulcerative colitis. And when I'm in my clinic, I hear about families like this all the time, where people will have a family history, but I also hear about where there isn't a family history. And perhaps we can talk about that, why that might be the case a little later on. There is a known family history, depending on which studies you read, anywhere up to a third of people with IBD. This is probably a better measure of what the risk is from a genetics. So what this is saying is that if you have a first-degree relative, that's a sibling or a parent, who has Crohn's disease, your risk of developing Crohn's disease is somewhere between 13 and 36 times the general population. So having a family member with IBD is the single biggest risk factor for developing IBD. And there's nothing you can do about that. That's in your genes, and we can't change our genes. As people say, you can choose your friends, but you can't choose your family. So you're stuck with your genes. If we think about what's going on in IBD in the world at the moment, is that we're seeing a rise in the development of IBD in newly industrialized countries. Historically, these have been diseases, the Western world, but now we're really seeing almost an epidemic of these diseases and other immune or autoimmune diseases in places like China, India, and so on, as they adopt a Western lifestyle, as they become more industrialized. Actually, the incidence, so that's new cases in the Western world, is sort of plateaued. It doesn't seem to be increasing so much. But we do have an issue, which is that because people with IBD don't have an increased mortality rate, they live as long as people without IBD, and the incidence is still going, and we've got an increasing aging population. We're getting more and more people in the West, but that's because we're just pouring more people with IBD into the tank, as it were, and we're not losing people with IBD. That's a good thing. That's a great thing that we're not losing them, but it does mean that we're just more and more people, and we're actually going to be having to deal with a new phenomenon soon, which is an increasingly aged population with IBD, and older people with IBD present different things for us to think about. These are many of the environmental influences in IBD, and people are working incredibly hard on this. I don't want to say that they're not. And we're finding out more and more things that can influence the risk of IBD. And you see some of these here. And some of these are really, really important, even in our clinical practice. Talk about smoking, we talk about diet, I talk about exercise, and dealing with stress and so on. Those are important parts of our clinical management. So why have we made advances in the genetics? Well, one of the things is that the cost of doing genetics has dramatically dropped. It's far exceeded this thing called Moore's law, which is that as time goes on, technology gets cheaper. You can see what's actually happened to the cost per human genome. It's dramatically reduced. You can now get your genome sequenced for about, well, depends who you talk to, $200 or $300. The problem is that the interpretation of all of that data and the storage of it, that cost has risen dramatically. So on my end, as a researcher, I'm spending much less on generating the data and much, much more on employing smart people to interpret the data. And that's an expense. That's a considerable expense. And that's one of the barriers for us for getting this into clinic. But we are, like everyone else, employing machine learning and AI to help us with this, because there are very, very huge amounts of data. And we're doing similar things with other data sets as well. So we've made great success in identifying genes for IBD. And out to 2020, we had more than, about 250 of these genes. That's what people say. What they actually mean is that we've identified regions of the genome which contain genetic changes which predispose to disease. And why do I make that distinction? Well, it's this. So again, what the name of the genes are is not important. But what this is showing is that we have a genetic variant, that's what this thing is, which is associated with Crohn's disease. And it's in a region of the genome, but you can see this region of the genome has all of these genes. And I can't tell just by looking at this which gene is actually driving this, or is there more than one gene? So even when we identify regions of the genome, we have a huge amount of work at each one of those regions to try and work out which of the genes is actually leading to an increased risk. But we have identified some specific genes. And one of the reasons for doing the genetics is it started to shed light on pathways and processes where changes lead to an increased risk of developing IBD. And these are three of them. Again, that it's not really important what they are. Though I will say that this one here, called adaptive immunity, has proved to be quite important in terms of developing new drugs. So some of you will know that we now have drugs which target this, IL-23. They're commonly advertised on TV. Certainly when I'm watching TV, they seem to follow me around. People that will know that there's a drug called ustekinumab or Stelara, and that targets this gene. So one of the reasons that we do genetics is to try and identify new drug targets. And we have some evidence that that's been the case here. What has the genetics told us? I think this is a better way of thinking about it. So this is the London Underground. And those of you who've seen me talk before will remember this, and I apologize. But I still think it's the best way of describing this. And what the genetics has told us is that if our destination is Crohn's disease, we all can start from different places, and different pathways or different lines on the underground can all get us to that destination. So you have the genetics in one pathway, the genetics in another pathway, genetics in that IL-23 pathway I was just talking about, but they all lead to this thing that we call Crohn's disease. And the same for ulcerative colitis. I'm just using Crohn's disease as the example here. So that's how these different pathways that we're identifying from the genetics play into the development of Crohn's disease. The other thing that we discovered from the early studies is something that we sort of knew from our clinical practice. So when I see people with IBD in my clinic, I always ask them about other autoimmune diseases. And I ask them about a family history of autoimmune diseases, because a lot of these genetic variants that predispose to Crohn's disease and ulcerative colitis also predispose to these other diseases. And here are some examples. And what this actually shows is the overlap between these various things. So if I take inflammatory bowel disease, for example, and this circle is the one that intersects with psoriasis, there's a lot of genetic variants that overlap between the two. That's the size of the overall size of the pie chart. But the really interesting thing is that some of the genetic variants seem to point in the same direction, but some point in the opposite direction. And we don't understand why that is. And I think that once we understand that, we'll understand a lot more about how the genetics is causing these diseases, because that didn't make sense to us to start with. But what it may be is that the genes are their expression, how much they actually function, are different in different tissues. So in psoriasis, some skin things specific to the skin, the genetic variant may have an influence one way, and in the gut, it may have an influence the other way. We don't know yet, but there's a lot of work going on to try and understand that. We also had some surprises with the genetics, and these are two studies which reflect work from our own group. We identified this gene with a very catchy name of SLC39A8. Again, do not remember this. What this gene had previously been associated with was hypercholesterolemia, hypertension, obesity, Parkinson's disease, and schizophrenia. And we actually identified that this same genetic variant was associated with Crohn's disease, but not ulcerative colitis. And there's another gene called LRRK2, which has a very strong signature in both Crohn's disease and Parkinson's disease. And we're now studying the role of the gut in developing Parkinson's disease and the microbiome because of findings like this, and trying to understand the overlap between Crohn's disease and Parkinson's disease. We also discovered that some of these genetic variants have been associated with infections in the past, such as leprosy and mycobacterial disease. Mycobacterial disease, the one that a lot of us know about, is tuberculosis. And I will come back to that a little bit later. The other thing that we discovered is that most of the genetic changes we were seeing that predisposed to Crohn's disease also predisposed to ulcerative colitis. And so something comes along and may push people one way or the other. And this explains why when there's a history of Crohn's disease, there's an increased risk of family history of ulcerative colitis and vice versa. These two diseases seem to go along together in families. And why one person may develop Crohn's disease and another ulcerative colitis, we don't really know. One of the biggest risk factors we do understand is smoking, and smoking has this opposite effect. If you smoke, you're more likely to get Crohn's disease. If you smoke and then give up smoking, you're more likely to develop ulcerative colitis. In fact, smoking protects from ulcerative colitis. That is not something that we would ever encourage people to take up to try and prevent ulcerative colitis, but it's an interesting sort of epidemiological finding. And these genetic variants might predispose to both, but they can have different size effects. And I'm going back to that IL-23 receptor, and what this shows in this plot is the width here in Crohn's disease is bigger in for the IL-23 receptor in Crohn's disease than it is for ulcerative colitis. So this gene predisposes to both diseases, but it has a bigger effect in Crohn's disease than it does in ulcerative colitis. And again, that might be important when we think about therapies and so on. Do they have a bigger effect in one disease than the other? So that was sort of the early studies. So where are we now? So we've discovered more and more genes or regions of the genome. And I think by the end of this year, we'll be well over 300. We're discovering new genes all of the time. But there is a big problem. I've already highlighted one. And one is that the vast majority of our studies are in people of European ancestry, white people. And our studies are lagging behind hugely in non-European ancestry populations. And we're not alone in that. This is true of genetic studies everywhere. Now, my group has been particularly passionate about this. And I want to highlight Tarlene Harutunians and Daylin Lee, who are both professors in my group. And they've been at the forefront of publishing studies like the ones I've listed here so that we can close this gap, because we think this is very, very important from a social perspective, from an ethical perspective, but I'm also going to show you why it's important from a scientific perspective. Now, what have we discovered? Well, we've discovered that some of the genes that predispose in one population do not in another population, but a lot of them do predispose in both populations or all populations. The effect size might be different between them, but what they're really pointing to is those same pathways that I showed you with my model based on the London Underground. It's not that we're discovering vastly different things in the different populations. They're pointing to the same pathways, though the genes might be slightly different. And this is just to emphasize, you can see here, this is genome-wide studies, and this is outside IBD. This is just across all diseases. And you can see two things. One is that in 2009, 96% of all samples that were in studies were of European ancestry. Now it's got better to 2016, but we're still failing, really. And you can look down here to see the density of where all of these studies are. And you'll see that the UK does a lot of these studies. So does the US, but there are virtually no studies in Africa and South America. And one of the important things that we're discovering now as these diseases develop in non-European populations is that the non-European populations have different clinical presentations. And that might be very, very important to try and understand the genetics underlying that. So we wrote this opinion piece that was published earlier this year, saying that we really, really needed to address these issues. It's true of research, but it's true across many of the different areas in science and medicine. So this is a big issue from an ethical, as I said, and social perspective, but there are very good scientific reasons for doing this as well. So I'm going to step out of IBD and talk about cardiovascular medicine and hypercholesterolemia. So a study was published a few years ago which identified this gene, PCSK9, as carrying a genetic variant in African Americans which protected them from developing high cholesterol. And this was a very novel and interesting finding, but the genetic variant was only found in African Americans, not found in European ancestry populations. But a company took this on and they developed antibodies to this. So they developed a drug to target it. And this drug has a dramatic effect on your cholesterol and is now one of the backups to people who can't take a statin for measuring for lowering their cholesterol. And it works in every population. So even though the genetic variant was only discovered in African Americans, this has had widespread benefit across all populations. So this is one of the good scientific reasons for doing these types of studies. I'm going to talk a little bit more about genetics across different populations because I want to show you another example of this. But I'm one of the things I do outside my research is I love history. I absolutely love history. And so when I get to combine sort of history and genetics, it really hits a sweet spot for me. And one of the things is to think about migration of populations. And of course, some of those have been enforced, like the slave trade and so on. And some of them have been the sorts of natural disbursement of humans out of Africa. And all of that is particularly relevant to populations in North America. So if you take populations in North America and South America, so African Americans are, you can see down here, are a mixture of African ancestry, so that's in the yellow here, and European ancestry. And some African Americans have much more European ancestry than other African Americans. Puerto Ricans, and we particularly study Puerto Ricans in our group, are a mixture of European ancestry, African, and Native American or Amerindian. And again, the European ancestry component varies a lot. If you go down to parts of South America, there's much more Amerindian or Native American components. And that is because, of course, the original Native Americans we believe came from Asia, went across the Bering Strait, so across from where modern day Russia, through Alaska, then down through America, and so on, and then we'll push further and further south. But this actually does have some implications for clinical practice. And this is another way of thinking it. This is a three-dimensional film. I hope you're seeing this, of when we plot people by their genetics, they can form in these clusters. And this is very, very important for when we do genetic research, that we understand this, because we need to incorporate this into our findings. But it's a little bit more complicated than that, and I'm really sorry to go into the genetics here, but I hope this movie makes it clear. So if you have parents who come from different populations and they have children, the children are a mosaic of their parents. We know that. But what actually happens is it's not this simple that they just divide their chromosomes like this. The chromosomes, as you go through generations, are mixing and mixing and mixing. And you can see what happens here as we go through the generations. They become a real mosaic within the chromosome as well. And so each one of us has a part of our chromosome which can be traced back to a different part of the world. So those of you who are European, and you would say to me, because I always ask people in the clinic, where are your folks from? I'm a geneticist. I really want to know that stuff. So if you say, oh, I might be, part German, part Irish, and so on. If we looked at your genome, we actually may be able to start pull out parts of your genome which are German in origin and which may be Irish in origin, and so on. And this is what your chromosomes actually look like. So each one of these is a chromosome 1, right up to the 20s here. But different parts of your chromosome you've inherited from different parts of your ancestry. And if you look, I just want you to look at the across the top here. These are our Hispanic studies. And we work with groups in Puerto Rico, Miami, and then our own Southern Californian Hispanics. And what you can see is that the Southern Californian Hispanics have much more Native American ancestry than their counterparts in Miami and Puerto Rico. So they're much more Amerindian and perhaps get that ancestry from East Asia. Now, why might that be important? Well, for example, if we go back to that IL-23 receptor, the one I told you where there's a drug now, it looks like that has a big effect in the European ancestry, but no effect at the African ancestry and no effect in the Native American ancestry part of your genome, which is really interesting. And there are drugs here, and one of the questions we're now asking is, does this influence whether you respond to this drug or not? Or is it like that PCKS9 example I gave you? It just pointed to the biology and this actually doesn't matter. We need to know that. So we're working on that at the moment. And one of the other things that we study is Ashkenazi Jewish genetics. And we found a very interesting thing when we looked at the Ashkenazi Jewish population here at Cedars compared to the European ancestry non-Jewish population, that there were subtle but significant differences in the processes that were going on in the genes that we predisposed to Crohn's disease in the Jewish population. And that was all genes that had previously been associated with intracellular bacterial response, whereas in the European non-Jewish population, it was response to fungi. So why is this important? Well, there are some drugs that are come out of this pathway, and there are drugs being developed in this pathway, and maybe they have differences between the two populations. We don't know. These are the sort of studies that we need to do. But I think this is, again, where history collides. There's a fascinating study which showed that if you look up here, one of the genes that's implicated up here in the Jewish ancestry is called NOD2. You can see it up here, this one here, and genes like it. And what this shows is the frequency of NOD2 variants, and this shows the burden of the Black Death in the Middle Ages, the plague. And what this paper suggests, and has some very nice data, is that the reason these genetic variants are in our population today is that they protected our ancestors from various diseases, including the Black Death, which, as many of you will know, wiped out huge populations in Europe. I mean, really, the European population shrank very immensely after this. And the people who carried these NOD2 variants may have been protected, and therefore because they were protected and the population shrank, the frequency of those NOD2 variants increased in the population. So this might be the price. Crohn's disease may be the price for some of these things in the past. I just want to highlight one thing I'm particularly proud of is that this year we actually founded a consortium, and I went down to Zambia to start studying the emergence of these diseases in sub-Saharan Africa, because we're now beginning to see IBD in emerge in sub-Saharan Africa as a Western lifestyle and they become more industrialized. So you can I said I'd mention briefly about bacteria and so on. It's not really the topic of this conversation, but we know a lot more about bacteria, good bacteria, bad bacteria. We don't really like to use that term. But I just want to emphasize that just like genetics shouldn't be looked at alone, we shouldn't be looking at bacteria alone. There's also differences in viruses and fungi in the gut. And just to give you an example of this, so we did some work with one of my colleagues here, and we identified that there was a fungi in the gut in some people with Crohn's disease, but it all depended on whether they carried a genetic variant or not. So here's the genetics and the microbiome colliding, and now we're doing a study treating people who carry this genetic variant with antifungal agents to see if it improves their Crohn's disease. So, this is an example of personalized medicine, and I think this is where the science is going. This fungi causes dandruff, and this was reported by the Daily Mail. This is the only time I've appeared in a national newspaper in the UK. Unfortunately, it was the Daily Mail. Those of you who don't know the Daily Mail, is sort of the equivalent of the National Enquirer. So I wasn't that delighted, and I definitely wasn't delighted with the way they told the story. But nevertheless, the science is very interesting. And I just want to say the other thing that Talian Haritunians in my group is leading internationally is looking at sex or gender specific effects in IBD. So we have a whole program around that. And it's not just related to pregnancy and IBD, but that's very, very important. But it's thinking about differences in the clinical presentation, differences in the genetics, differences in the immunology, the effect of hormones and so on. So what does it mean for the clinic? What does all of this stuff mean for the clinic? Well, I'm just going to give you a few quick examples of that. I know that time is going on. So can we use the genetics to test for IBD? I'm asked this all of the time. And some of you will have seen this data before, but the story has not changed. The short answer is no, and I'm going to show you why. So if you can send off a swab or a spit into a tube and send it off to a genetics company, they will tell you what your risk of Crohn's disease or ulcerative colitis is. But if you read the small print, they can only tell you it ranges between 0% and 1% based on your genetics. That is not a good test. That is not a good test at all. And in fact, when someone looked at this and they looked specifically at Crohn's disease in 5 different people across 2 companies, they got different results in 3 out of the 5 people. Again, horrible test. And so Francis Collins, who was head of the NIH at the time, just said this is the sort of the modern version of genetic snake oil. That's not the end of the story. So I want to introduce you to this concept of polygenic risk scores. What it really means is we should actually not be looking at individual genetic variants. We should be looking at the whole genome, because I've told you there are 350 areas of the genome associated with developing IBD. And what you can do is you can start to add up all of these genetic variants and create a score, a genetic score of what your risk is. And so once you do that, you get what we call this binomial distribution. And so you have a small number of people who have a moderate number of people who have an intermediate risk, small number of people have a high risk. And this is now beginning to be used across science. So here I go again out of IBD, and I want to talk about cardiovascular disease. So this is coronary artery disease. And what this shows over here is your risk of developing coronary artery disease is increased with all of the things we know: smoking, diabetes, family history, if you're obese, hypertension, high cholesterol. That's all of these things. But look, if you have a high genetic risk score, your risk is higher than these things. And if you put the genetic risk score together with these other things, then you can really identify people at very high risk. And what we're identifying is people at high risk of coronary artery disease who don't have any of these risk factors. So it's beginning to show us people who are at high risk without us understanding the risk factors, which is very interesting. And of course, if you think about risk of coronary artery disease, one of the things that there are all of these things that people do: smoking, poor diet, drink too much, lack of exercise, and so on. And these are risk factors that we sort of accumulate through life. But our polygenic risk score, we know at birth. We can know at birth. So we can actually put people in, give them an evaluation of what their risk might be at birth. And so perhaps you can then target interventions and so on to people. Now, there are ethical issues associated with that. I don't want to deny that, but this is something that we need to think about. And another great example of this is breast cancer. So everyone knows about the BRCA mutations, the breast cancer mutation genes. If you carry those, you're at increased risk of breast cancer, ovarian cancer, actually prostate cancer as well. But your risk is modified by what's going on the rest of your genome as well. So some people carry those mutations and have a very high risk, but some carry the mutations and have a risk which is similar to the rest of the population because the rest of their genome somehow protects them from the effects of the BRCA mutations. So this is where the genetics is going. Sorry to any of you who are eating. I should have probably warned you before I put up this slide, but I usually try and put it up halfway through a talk because it sort of wakes people up a little bit. When I sit in my IBD clinic, I'm faced with a variety of different problems to try and deal with. So no two patients are exactly the same. And some people have very severe disease. Some people have mild. Some people respond to therapy. Some don't. Some people have disease in different parts of the gut. And some people have disease outside the gut, skin manifestations, and so on. So one of the things that we're thinking about is actually how many diseases are we dealing with here? And we're trying to work to try and understand the genetics underlying these different diseases. And it's always worth remembering and going back to history. So Burrell Crone and his colleagues described Crohn's disease in 1932. They weren't the first people to describe it. That was a British doctor. I just have to get that in, but it's become Crohn's disease. And they described this disease of the small bowel, which is a lot of what we see with Crohn's disease. Now, a British surgeon then identified similar changes in the colon, so the large bowel, wrote to Burrell Crone and said, you know that disease you described in the small bowel? Well, I've seen it in the large bowel now as well. And Burrell Crone wrote back to him and said, no, I don't think this is the same disease. So even though we call it colonic Crohn's disease now, Burrell Crone did not recognize it as the same disease. And perhaps he was just way ahead of his time, because those polygenic risk scores I was just talking about, when you look at them in IBD, here is small bowel Crohn's disease, and here is colonic Crohn's disease. And colonic Crohn's disease is actually much closer to ulcerative colitis than it is to small bowel Crohn's disease. So this is one way that we're thinking that we're going to move on from this these diagnoses of Crohn's disease and ulcerative colitis to a much more molecular-based classification, which is what they're doing in oncology and cancer. They're stopping describing or thinking necessarily about the cancer, about the location of it, and more about its molecular signature. So you can have a drug that may be very good for certain types of breast cancer, also very good for certain types of, say, pancreatic cancer, but then other drugs for the different types of cancer in those locations. So that's an idea that we're trying to adopt. And we know clinically that there are big differences between small bowel and colonic Crohn's disease, and also that the drugs that we use, if you look at all of the clinical trials, people with small bowel disease do not do as well as the people with colonic disease. So we need to understand what's going on here in small bowel disease so we can develop new drugs to target that specifically. And we're not alone thinking about this. You know, the other immune-mediated diseases are thinking in the same way. So just like the cancer example I gave you, we could think about, we have a molecular signature of Crohn's disease, which is similar to some forms of psoriasis. The drugs in those two groups work well for both those, but there may be other Crohn's disease where the drug doesn't work and other psoriasis where the drug doesn't work. So we're trying to move away from the thinking about moving away from this classification that we use at the moment. So what about predicting the future and so on? So we try and take a very holistic approach to this. And here's work which we did where we looked at genetic variants which are known variants that predispose to developing a thrombosis, so a deep venous thrombosis or a pulmonary thrombosis. Now, the reason this is important is these are the biggest cause of death in people with IBD. So people with IBD are at increased risk of this, and this is something that we're always worried about. And what we found is that if you took those individual variants that we know that our hematology colleagues will screen for and looked at the risk of developing venous thrombosis. If you carried one of those, your risk is about 2.5 times. But if you ran the rest of the genome and pulled out all those hundreds of variants which have a very small risk and added them up and created a score, and you had a high score of that and this, your risk of a venous thrombosis was 8 times that. So when we're thinking about the genetics of IBD, we're not just thinking what's going on in the gut. We try to think holistically. And here you can think about venous thrombosis, but the same is true of cardiovascular risk. The same is true of cancer risk and so on. Because once you have that genetic data, you can think about all of these things. We've even got a study looking at the risk of depression, because we know quite a lot about the genetics of depression in the general population. We're applying that to our IBD population. Another way of thinking about it is, can we make our drugs safer? Any of you who either yourself or family members have taken either azathioprine or mercaptopurine will probably have had a test done beforehand to check that you didn't carry a genetic variant in this gene, TPMT, because 1 in 10 people carry one variant, 1 in 300 carry 2 variants, and if you take the drug at normal dose, you're at risk of a life-threatening complication if you fall into this sort of 10% group. So these genetics have helped make the use of these drugs safer. But we worked very closely with a group in Korea, and what they observed was that Koreans were at high risk of developing this complication, but had lower levels of that genetic variant that I just showed. And they discovered a completely new gene associated with this bone marrow toxicity called NUDT15. Now, I work just up the road from Koreatown. I have quite a few patients of mine who are of East Asian ancestry. And so this data would suggest I shouldn't be testing for that other gene. I should be testing for this gene in that population. It is rare in Europeans, but if you have it, the genetic variant, then you're more likely to get that bone marrow toxicity. The really fascinating thing is, do you remember I was telling you about the Amerindian component of the Hispanics? That because they're descendant from East Asians, the Hispanics I see in my clinic are much more likely to carry this genetic variant than my colleague in Miami who's seeing her Hispanics. So the Hispanics in Southern California, we should be screening for this genetic variant before we use this drug as well, because of that ancestry from East Asia. We've also identified a genetic variant which very strongly predicts whether some people will develop antibodies to the drugs infliximab and adalimumab. And this is another really great example of where genetics can influence clinical practice. Because if I know that people are carrying this genetic variant, I want to be very careful about using these drugs, and I probably have to either dose them much higher, or I have to think about adding in an additional drug to protect them from developing those antibodies. So someone asked me about new drugs, and the genetics can point to that. And I've given you this one example before. This IL-23, we discovered this genetic variant was associated with Crohn's disease. It's associated with a number of autoimmune diseases. And now we have these drugs coming through in the clinic which specifically target this. And the genetics is pointing to lots of different things, but the genetics can point to something else as well. We see this very unusual phenomenon sometimes, and some of you may have experienced this. Anti-TNFs are given to treat Crohn's disease and ulcerative colitis and also psoriasis, but a small percentage of our patients develop psoriasis with the drug, and we don't understand that. And part of this may be the genetics. And I remember I was talking about those genetics that work in different directions in those in the different autoimmune diseases. So here's an example of a gene which has a risk for ulcerative colitis and asthma, but it protects from some of these other autoimmune diseases. So what you can work out from this is if you developed a drug and you treated your patients with ulcerative colitis and it worked, they may be at risk of developing psoriasis as a side effect. So this is another way that the genetics can help us in drug development because it can help us predict what side effects people may be developing. And I think this is going to be a very big story going forwards. One of the problems that we have is we say we have all of these drugs, but if you really look closely and we look at all of the different drugs that we're using at the moment, if you look at overall remission rates at week 54 after starting one of these drugs, it's about a quarter of patients in some of these studies, maybe up as much as a third. We have a at least two-thirds of our patients who are not responding to these drugs, and we have no way of predicting that. It's a question of, we have this drug, I can talk to you about the side effects of it, but I don't have a marker that says you can respond to this drug. So we've been working on that, and this is work come out of Cedars, where we've started with a genetic variant. We did some machine learning to try and understand the genetics around the protein that it produces. And this was this gene, TNF superfamily 15. Again, it doesn't matter what it is, except it works in both inflammation and fibrosis. That's those narrowings in the gut, the strictures, and so on. So it has double action, which is pretty unusual. And we developed a drug, and the drug seems to work in both ulcerative colitis and Crohn's disease. And the data are pretty impressive. But what was really interesting is because we started with the genetics, we believe that we've got a marker or a set of markers that will identify people who are more likely to respond to the drug. And that's precision medicine. And that's not really been done in autoimmune diseases. Our colleagues in cancer are doing this all the time, but this would be one of the first examples outside cancer where we have a precision medicine approach to these drugs. So finally, and I know that I'm really butting up against time here, what about cures? So we do have examples of cures in IBD, and this is where we have these quite rare at the moment examples of what we call monogenic forms of the disease. And this is young children who develop IBD in the first couple of years of life, first 2 years, sometimes say first 6 years, and they have very severe disease. And the reason they have it is that they have often they have what we call knockout of a gene. So they'll inherit from each parent a rare mutation which stops the function of a gene. And when that happens, that gene actually causes IBD. You don't need anything else. You don't need any of those environmental factors. You don't need other genetic variants or anything. It actually causes the disease. And if you can work this out, you can actually correct that. So in this poor kid's case, who had very severe disease and also joint inflammation, skin inflammation, they worked out that this was all coming from bone marrow. The kid got a bone marrow transplant and it cured them from their IBD. And now we started right back here with just identifying a few of these, but now we're identifying more and more of these genetic variants and more of them where we think that something like a bone marrow transplant might be curative, or we know exactly what the gene is, so we can target that protein. So it's like, goes back to the oncology things. You have, this is the problem, this is the fix for it. So this is very, very personalized medicine. And what we're doing now is because we've got genome sequencing in all of our IBD patients, we're trying to make the case that there are people who develop disease much later in life, for whatever reason, who follow a similar signature to this, and that we can perhaps target these sort of type approaches to a greater percentage of our patients. And so bit by bit, start to think about how we can cure these diseases. And when you look at the genome, there's one study that shows that 63% of IBD patients have what we call actionable variants. Now, they're not all IBD related. Some of them are, some of them I've talked about, but others are breast cancer mutation or hypercholesterolemia or so on. But that goes back to what I was saying. We really like to think about the holistic effect, the holistic approach to our IBD patients. And the other way we think about this is to think about cure. And there are some great examples of studies going on, and one is just from the Department of Defense. So if you're in the Defense Forces, there's no consent involved, but you have to have a mandatory screening for HIV and other infectious diseases every year. So they take a blood test from you, check that you don't have HIV, and then they stack the blood samples, because they never throw anything away, in these warehouses. These are all blood samples from people taken over the years. Now, as you can imagine, it's people in the armed forces are relatively young. And as time goes on, some of them will develop IBD and other diseases. So you can actually go back in time and look at their blood results or their blood samples, and you can actually identify things in the blood samples that predate their diagnosis of IBD. And this is what some groups have been doing. One of my colleagues here is doing it. These are some of the serological markers, but we're now thinking of adding in the genetics onto the serological markers to see if we can identify people at very high risk of developing IBD, because this is where we may have some real benefit with those preventative strategies, microbiome intervention, changes in diet, maybe keeping a very close eye on these people. So if some of their inflammatory markers start to go up, we can intervene and so on. And people with other autoimmune diseases are already thinking about giving medications which are safe to people at high risk to see if they can prevent the onset of this. And they've shown some benefits in rheumatoid arthritis, and a paper just came out this week in type 1 diabetes as well. So I think this is a very exciting space going forwards. And finally, how do we put this all in the clinic? Because it's very complicated. I've only talked about genomics, there's microbiome, there's transcriptomics, proteomics, with machine learning, artificial intelligence, all gets very, very complicated, huge data sets and so on. So we're working on strategies so that we can actually distill this down. And you see here a couple of figures where this is the risk up here of someone developing complicated Crohn's disease out to 3 years after diagnosis, compared to this individual who has a much lower risk. So in theory, you would take a very different clinical approach and give different advice as to what interventions you should take for this individual compared to this individual. So we really need to distill it down once we've discovered the things that we think are important. So that's all I had to say. And I know that I've gone on, but you give an Irishman a chance to talk and they will just keep talking, particularly about subjects they're passionate.
It was very interesting, actually. So it was fantastic. Does anyone have any questions? I have a million questions, but I don't have to ask them all. But does anyone have a question? And Betty, I know you had some questions. Dermot answered a little bit, I think, of what you were asking about the new medication, I think. Was that what you were asking about, Betty?
Yeah, he addressed that. Yeah, and also he addressed 2 of my questions.
Mute that person. Okay. Does anybody else have a question?
I saw someone's hand up.
Oh yeah, Julie.
Yeah, I was just wondering if okay, so my son has Crohn's disease, and I'm just wondering if you, from a clinical perspective, not able to determine the cause of elevated liver enzymes and associated symptoms. And it was interesting because I've always wondered about his dandruff, that he always has a hard time controlling. I was just wondering, would you suggest genetic testing for, I don't really, it's sort of like we're flailing a bit, and I'm just wondering.
Yeah. So there's no indication for genetic testing at the moment. Some of the stuff I've shown you today, and I should probably have said this right at the beginning, is really where we're trying to push the science. So a project that we're doing is we have about 10,000 people that we see at Cedars with IBD and have seen. So we're looking at every single person's genome to try and pull out the information so that we can then go to regulatory people and say, look, it costs $400 to do a genome. If I ran a panel of bloods like a CBC and a CMP and a CRP and so on, it costs $1,000. How many times do we do that? For one off $400, we can get the genome sequenced, and then we have that data ready to go on everyone. The problem is if you get your genome sequenced at the moment, it's not in that program. It's not people pulling out the variants that are important, and so on. I'm not encouraging people to get their genome sequenced. Personally, I am encouraging people to join up to research projects, because without that data, we're not going to be able to persuade people.
Thank you. I also just wanted to know about the founder effect. So you did, I'm just curious, like I know for Ashkenazic Jews, there's the founder effect. Is that even a valid theory anymore?
Oh yeah, no, it is. It
Okay.
is. And I sort of alluded to it with that Black Plague thing.
Yes.
So we study two populations which have a founder effect, really. The Ashkenazi Jewish population, because of persecutions and so on, was squeezed down to a very small population and then is expanded. And when that happens, you get rare variants that happen to be in that population, and then they, as the population grows from that small population, those rare variants are higher frequency than you would see in other populations. So that is definitely true of the Ashkenazi Jewish population. And the other population that's true is the Hispanic population, but the Amerindian component of the Hispanic population, because the Amerindian, the Native American population, has had, similar, very significant shrinkage in size because of persecution. And I'm not going to go back into history, but everyone knows the reasons why. So, yeah.
I know does anyone else have a question? I was wondering, actually, because you said, it was really interesting when you said that the biggest risk factor is the genetics. And I feel like, since Lowell's had, been dealing with this for a long time, I feel like that was not something that was said. Is that like a newish thing? Because they didn't really know that was the biggest risk factor.
Yeah, it's the single biggest risk factor, but the way I explain that in the clinic is, people often say to me, so I've got Crohn's disease, what are the chances of my son or daughter having Crohn's disease? And the answer is that there is an increased risk, but actually the way to think about it is that they have a 90% chance of not having Crohn's disease or ulcerative colitis. It's just higher than the general population.
And then what about when you're talking about the genes? I feel like at some point, not in this talk, but you had said something about the genes that they're like people who have Crohn's or colitis, that there is like a gene that the majority of Crohn's patients will have, or a couple, and then there's the outlier genes. So like, is that correct still, even though you've discovered more genes?
Yeah, in fact, if you take those monogenic forms, you would find that those genetic variants in like 1 in 10,000 people, that genetic variant. If you take that IL-23 receptor, 99% of healthy people carry that variant. It's 93% or 90% in Crohn's disease. So you see the full spectrum of the genetic variants. And that's why I'm saying this, that what we've really discovered recently is that you need to really start thinking about the whole genome here. You need to be putting all of these things together.
Okay. And then this is like the $1 million question, like, because I know we've talked about precision medicine and personalized medicine, and I know there are some things that people are doing now, like being able to test for the antibodies for Remicade, but like, it doesn't seem to have translated very quickly to the patient population, all of these things that are being discovered. And I know you touched on it at the end, but I think it's frustrating for people to like not to know this information and then not, be able to understand how that's going to affect them or how that can be
Yeah.
used to optimize their therapy or their disease, manage their disease.
Yeah, I think that the way I like to think about it is that, there are very few revolutions in science. It's very much evolution. And the way we practice IBD now compared to 10 years ago is actually transformationally different. But it's been incremental in terms of the studies coming out and so on.
Mhm.
And a lot of that is down to more personalized medicine. But personalized medicine means different things to different people. So I showed you that example of what we call a companion diagnostic, which might tell you you're more likely to respond to that certain type of drug. That's sort of the holy grail, and we're just beginning to make progress there. But we've really made progress in discovering some of those genetic variants associated with toxicity with some of the drugs, with the antibodies to the drug, and then also antibodies to infliximab and adalimumab, or Remicade and Humira.
Mhm.
But also the personalized medicine is also around individualizing drug dosing, which we do now, which we never were doing before. And then having these additional mechanisms as well. And now beginning to think about what ways do we distinguish which medications people should go on. I said we don't really have ways of thinking about that, but there are some ways. So some of the drugs we know work in people who have arthritis as well as their IBD. So we would then choose a drug based on that. And that's a form of personalized medicine. So we're getting there. We're definitely getting there. And I know it can seem like it's slow, and to a certain degree it is slow. But then occasionally we have to sort of look back 10 years and go, wow, I can't believe how far we've actually come in that period.
Yeah. No, I mean, I look back 20 years and I think just even the way that the disease is monitored and there has been a lot of progress, and I know that like at Mount Sinai, their team did a lot of that with testing to see who might have like skin reactions to Humira and being able to maybe change the dose or have to switch therapy. So I know it's going on. I know it's being utilized more and not just being talked about. Does anyone else have any other questions? I wrote down so many things, but I don't need to ask all of them anymore.
I have a question.
Okay, Janet.
Hi. Hi, Dr. McGovern. I think years ago in a lecture you gave, I think in Thousand Oaks, when I invited you, I think you said at some point you were going to look at patients who had Crohn's and psoriasis, however you pronounce that.
Yeah.
And you were going to start classifying patients in different ways so that it would be almost like the way hepatitis has hepatitis A, B, C, and D. You would have patients, Crohn's with psoriasis, Crohn's without psoriasis, Crohn's with this, Crohn's with that. Are you going to get to where you classify patients and a patient will know that they have Crohn's A,
Yeah, that's really what I was pointing to with that molecular scoring system, and that was just based on disease location. But we're also working very hard on people who have arthritis, psoriasis, people who have non-response to anti-TNFs, for example. So we're trying to incorporate all of that and come up with a much more molecular classification than we use at the moment. We use very blunt tools at the moment with ulcerative colitis and Crohn's disease. It's just that they're just really not fit for 21st century medicine.
Also, I thought another time when you gave a lecture at UCLA, I think it was, I believe you said something like, soon it would be possible to have patients spit on a slide or something and try different medicines on that slide, and you could know which medicine would work for the patient without them having to try it for a few years and then try another one for a few years. Is something coming? I know in my case, my son is still trying to find the right medicine after all these years.
Yeah.
is there something coming so that we could classify the patient or somehow get them closer to knowing which is the right medicine? Go ahead.
Yeah, so I think what I said was that we may be able to take a saliva swab or spit into a tube, run the genetics, and choose which medicine. And that's the example was that drug that we've developed, because we started with the genetics. So you may be able to take that genetic test and you're more likely to respond to that. And that was true in the phase 2 studies, which are relatively small, and the phase 3 studies are now being run. So we'll have the data soon to confirm that, or not, that that's the case. The other way that we're thinking of predicting whether you respond to medicines or not is using our big databases to say these factors are associated with response or non-response to any class of drugs, anti-TNFs, whether it may be. But the final way we're doing it as well is using organoids. So there we can get a sample like a blood sample or whatever, and we can create mini guts with little immune systems, and then we can provoke them to sort of simulate inflammation, and then try the various drugs on them and see if it damps down the inflammation, or which drug damps down the most. And that would be another way. And I think that is somewhere where we're going in the future. Our colleagues in cancer are sort of doing that. I'll give you one example. So they have been able to create not mini guts, but mini hearts that beat. And they then expose those hearts to some of the cancer therapies, because some of the cancer therapies cause cardiac toxicity. And they're using that sort of technology to try and predict which drugs that they should give to people to try and avoid that cardiac toxicity. So those are the sort of ways that we're thinking about trying to help decide which drugs people should go on.
And one last question, if I may, is let's say you find some really good, helpful tools and methods to diagnose patients that are different. How long does it take to get from you to the doctors in the various neighborhoods of the world?
Yeah. So we're very conscious that we need to make sure that we citizenize and democratize any advances that we have, both to our colleagues working in private practice or outside academic medical centers, but also to all populations. That's why, I was stressing the importance of the non-European ancestry populations. And there are ways that we can do that. One is to try and make the tests as simple as possible. So with this drug, it is simply get a swab, send it off, get the genetics done, and you almost come back with a traffic light sort of system, which is go, no go with this drug. So we try and make it very simple. The other way is to produce the thing I showed right at the end, which is that little thing that can sit on someone's phone, plug the data into it, or absorbs the data, and it will give you a prediction model of how things will go forwards. We're very conscious that we can sit in our ivory towers, do very complicated things, come up with algorithms, use our AI and stuff like that. But if it's not helpful to someone who's working in the community, then we might as well not be doing it. So we're working very, very hard to try and make sure we cross that bridge.
Thank you. Is there anything we can do to help as civilians, people not in the sciences?
I think that helping us with our research by, if someone asks you to donate samples or whatever, that's incredibly helpful for us, because we can't do it without that. Yeah. I'm seeing some questions in the chat.
Yeah,
so
can you answer? I know Tori is on the East Coast, I think, so it's probably late there. So maybe you could answer her questions.
So are there other centers using? Oh, answer that one first. Oh, she asked two. Okay. Yes, lots of other centers. We're working very closely with them. This is a team sport. Internationally, we're working with them. We're trying to develop a new classification for IBD, and I'm working with centers all around the world to do that. When do we think the CEDARS drug will come to market? Well, it has some hurdles first. So phase 2 studies were very, very successful. Phase 3 studies are ongoing at the moment. They should complete recruitment sometime next year. If they're successful, then they'll have to go to the FDA and get approval. So I think we're looking at towards the end of next year, maybe even right at the beginning of 2026. How long does the precision medicine process take for someone who's newly diagnosed? I think that there already is precision medicine for people who are newly diagnosed. I would hope that your clinician is thinking about what sort of disease you have, what the prognosis is, therefore, what is the personalized approach to managing your disease. Now, that's based on existing things that we have. Do you have these extraintestinal manifestations? Where is your disease location? How is your disease behaving? That sort of thing. In terms of the genetics, there's some things already. I mentioned the toxicity and the immunogenicity thing, and that's already available. People can use that. But the other stuff is up to people like our group to prove to payers that we need to get genomes done in everyone. And we're working incredibly hard to do that. And we're not alone. We're working with other people. So I'm hoping within a couple of years, we would be able to persuade, with the costs of the genome coming down, that we would be able to persuade them that we should be doing genetics in everyone.
Okay. Thank you. Can I just ask like one or two more questions? It's about the monogenic, I know the VO, IBD, and is the bone marrow transplant like very risky? Because that's what I thought.
Yeah, it is. I don't want to emphasize, overlook that. The thing is with those kids is that they don't have any other options.
Yeah.
They've run out of options. They do not respond to any other drugs, and they get life-threatening infections and so on. Now, so it almost becomes, the only choice.
Yeah.
Now, where I think this is going to be very interesting is, people have tried bone marrow transplant for treating Crohn's disease in other populations, people who've not responded to all of our drugs and so on. And actually, bone marrow transplant looks like it's quite successful for Crohn's disease. The problem is it comes with big risks. And so people have backed away from doing those studies and so on. If we can identify the people who are most likely to benefit from that, then it changes the risk-benefit discussion.
Right.
And that's where we're trying to see how much of those very early onset IBD signals sort of bleed out into the rest of the population.
Okay.
Yeah.
I just have one more question, because you talked about machine learning and AI, and I had met with this woman recently who is at a company, and they said that they used AI to develop, to find a pathway to treat IBD. And they tested it in Australia on healthy people, and there were no side effects. And then she's told me they're going to start doing trials, and that maybe Cedars is one of the sites. And this person was like the accountant, so she didn't seem to know very much about IBD. So I was asking her, like I was saying, do you understand there are different pathways or not just one? But she didn't really, she was like wanting to give me like hope, like, oh, this is going to be the cure. And I mean, obviously there's something to it because it seems like they've gained some traction, but do you know anything about that? And I think, I know when you talked about using machine learning, it's in a different way, but they talked about actually finding a way to cure or treat the disease.
I don't know which company you're referring to, but there must be about 100 companies.
They're called Athos Therapeutics. But
yeah, but I don't necessarily want to go into them specifically, but there must be 100 companies out there who've taken this idea of just applying AI and machine learning to data sets and so on. The one thing I would say is that I know we're sort of going through an AI revolution and everyone's talking about it and so on. But to be honest, we've been using AI in genetics and big data for a while now. it's and again, it's not something that's just changed overnight. We sort of been just gradually been using more and more powerful algorithms and so on. There's a big step between, and we used a machine learning approach to identify that diagnostic with the new drug, for example. But there's a big step from identifying something using that approach with data and then getting it into people and proving it's safe and so on. You have to do an awful amount of biology to understand and to prove that the signal does or is telling you what you think it is, because ultimately we're going to end up trying to treat people with this, and the last thing you want to do is end up the AI algorithm is incorrect, and you end up poisoning people. So it doesn't change that part of the drug development thing. It's a lot of work.
Yeah. Okay.
Yeah.
Does anybody else have any questions before we wrap it up?
Really interesting.
Thank you so much. It was really fascinating. I could listen to you talk about this stuff because it's really interesting and helps to like understand more about the disease for sure. And also to see all the advances that actually have been made through the research over the years. It's really encouraging. So
Yeah.
thank you.
Yeah, no problem.
Thank you.
And thank you
for your
everybody for
support.
joining.